Hi everyone. In this video, we're going to look at the final stage of cellular respiration, the electron transport chain, and ATP synthesis. If you haven't had a chance to watch them yet, I'd recommend that you pause this video and go back and watch redox reactions and the videos on the earlier stages of cellular respiration before you continue. The electron transport chain is the third and final stage of cellular respiration. Like the citric acid cycle, it is an aerobic or oxygen-requiring process, and it occurs in the mitochondria, with the proteins of the electron transport chain being embedded into the inner mitochondrial membrane. Remember from previous videos that mitochondria have a double membrane, with the outer membrane being smooth and the inner membrane being folded into cristae. This helps to increase the surface area of the inner membrane, allowing more electron transport chains to be embedded, and thereby maximizing ATP production capabilities. Let's zoom in to have a closer look at the electron transport chain in action. We'll start with our NADH, which remember we produce 10 of by reduction of NAD+ during the earlier stages of cellular respiration. NADH will feed its electrons into the first protein complex of the electron transport chain, becoming oxidized to NAD+ in the process. The energy from the electrons can be harnessed by the protein complex to perform active transport, pumping hydrogen ions into the intermembrane space, the gap between the inner and outer mitochondrial membranes. From here, the electrons will be passed to complex 2, which is also the stage at which FADH2 can feed in electrons, becoming oxidized to FAD. The electrons will be passed through the remaining components of the chain, with the potential energy being used to pump more hydrogen ions into the intermembrane space. Finally, the electrons will be handed off to oxygen, which will combine with protons to form water. This is the reason why cellular respiration is considered an aerobic process. Without oxygen to accept the electrons, the system will effectively back up and shut down, leaving glycolysis as the only means to generate ATP in the absence of oxygen. You might have noticed that we still haven't actually produced any ATP in this process. The potential energy from the electrons was used to set up a hydrogen ion gradient by active transport, and that is what's going to provide the energy for ATP synthesis by a process called chemiosmosis. Remember from when we looked at membrane transport that diffusion drives substances to move from an area of high concentration to low, and that ions, like these hydrogen ions, can't go directly through the membrane because they're hydrophilic and the membrane is hydrophobic. To facilitate the diffusion of the hydrogen ions, they're going to travel through a channel in an enzyme called ATP synthase. As they do so, they cause the protein to rotate like a turbine. And this energy can then be used to join ADP and phosphate, producing ATP. This can produce in the region of 26 to 28 molecules of ATP for each glucose that we started with, resulting in a total of around 30 to 32 ATP per glucose. Rather than substrate-level phosphorylation, which was used in the earlier stages of cellular respiration to produce ATP, chemiosmosis produces ATP by oxidative phosphorylation, because it is powered by the redox reactions of the electron transport chain, rather than from phosphorylated substrate molecules. So in summary, the electron transport chain and chemiosmosis together required 10 NADH and 2 FADH2 from the earlier stages of cellular respiration, along with 6 molecules of oxygen and 26 to 28 ADP and phosphates. From that, it produced 10 NAD+, 2 FAD, 6 molecules of water, and 26 to 28 molecules of ATP. Thanks for watching, and if you haven't had a chance to yet, please go and check out the videos on the earlier stages of cellular respiration.